Hello again, ladies and gentlemen. Now that we've talked about cells and the types of cells that we have and what can happen inside cells, now we're going to talk about biomolecules. Basically, these big macromolecules that make up the cell. Everything that is inside the cell is made of some type of biomolecule. All living things have biomolecules, which are basically these carbon-based structures. So you have your foldable in front of you, and we're going to go through each component and write our notes down. Here we go. So the first biomolecule we're going to talk about is a protein. And in order to recognize each one of these biomolecules, we have to see pictures and we have to be able to understand what's going on. So when we see a protein, basically, we're going to be able to tell that it's a polymer of a protein, the big part of a protein, if we see the peptide bonds. So, draw an arrow to one of the bonds holding these things together. This is called a peptide bond. Okay, so it's going to link all of these little pieces together. Remember, polymers are the big units and monomer are each of the single units. Now you're going to open up the flap of your foldable, and underneath protein, you'll see the other notes that we're going to write. So we're going to write stuff about the monomers, and we're going to give some examples of proteins. So let's start with the examples that we see. Some examples that we see of proteins are things like that are in the picture, like steak and chicken and eggs and fish and beans. Basically, all these things are meats or they are made of muscle. So all those things you use to move, those are all made of proteins. Or even enzymes. We're going to talk about enzymes a little later on in our review, but just keep that in mind that enzymes are a type of protein. So now let's look at the monomers of proteins. A monomer of the protein is going to be called the amino acid. So go ahead, write that down. The amino acid is pictured right below where you just wrote monomer. So let's annotate what we see in there. When we look at an amino acid, we're going to see this side group right here that is going to be our amino group. So label it amino group. The amino group is going to have the nitrogen in it. Always, we're also going to see this R group, which is basically this R side chain. And on the other side, we've got the C's, O's, and H's. This is going to be called our carboxyl group. CARBOXYL. And the whole purpose of a protein is to be able to provide structure, support, and do major cell functions. So we're going to write that down as structure, transport, and enzymes are the ones that help us speed up reactions. The second biomolecule we're going to look at is, if you close your foldable back up, we're going to look at nucleic acid. Now, nucleic acid, that is NA, right? So that is things like DNA and RNA. And it always, you're going to see this kind of double helix structure. So anytime you see that DNA molecule with its double helix, we know that biomolecule is going to be a nucleic acid. When we look inside this molecule, each one of these strands is held together by a bond. Proteins have the peptide bonds. Nucleic acids are going to be held together with hydrogen bonds. Now let's open up and look at the monomers and the examples of nucleic acids. So like I said before, the examples of nucleic acids are going to be DNA and RNA. If you see this double helix, that one is going to be your DNA. It is double. The single-stranded one that we see is going to be our RNA. And every single one of these, whether it's DNA or RNA, is going to use the same 4 nucleotides, A, T, G, and C. Adenine, thymine, guanine, and cytosine. Remember these 4 bases. A pairs with T and G pairs with C. Now let's look over here at our monomers. Those monomers of nucleic acids are going to be those ATGC. Those are called nucleotides. That is the monomer of a nucleic acid. Now, right below that is a picture of a nucleotide. So let's go ahead and label the main the main three structures we see. So the base, this middle part here, is going to be your sugar. For RNA, it's a ribonucleic sugar. For DNA, it's a deoxyribose sugar. Attached to it is going to be this P, this phosphate group. That's going to make up the backbone of the DNA structure. And on the other side, that is where these ATGCs come into play. This is going to be where you see your specific nitrogen base, whether that's the A, the T, the G, or the C. And the function of this DNA is to hold our genetic information. It's basically going to tell you how to code for your proteins, and it's going to tell you why you look the way you do. So the function is holding genetic info and instructions for making proteins. The last two biomolecules we have are all about energy, but they give us different kinds of energy. So let's look at carbs first. Carbohydrates, you're going to be able to distinctly recognize in a picture because they're going to have these 5 carbon ring structures. Every single one has 5 carbons, and they're put together in this ring-like structure. So here's a ring, and here's a ring. So the first thing we are going to see are examples of carbohydrates. So things like pasta, rice, grains, breads, those are our examples. All of these things include sugars. A carbohydrate is basically a sugar. The carbohydrate we're going to see in plants is that sugar called glucose after we've done photosynthesis. But in the things that we eat, it also includes things like bread and starches, things that are basically going to fill you up and you basically carbo load. Now, when you carbo load, that means you are trying to get all this energy kind of before an athletics game or something like that. Now let's look at the monomers of these things. We'll see that the monomer looks pretty much like the entire biomolecule. It's got this 5-ring structure to it. When we just have one of these, we call it a monosaccharide. These monosaccharides are going to do the function of giving us quick, available energy. It's basically going to be our primary source of energy. So if, like I was talking about, you wanted to run a race, you are going to want to eat pastas and breads, all these things full of carbohydrates that's going to give you this quick energy. Speaking of races, let's go ahead and race through this very last biomolecule, which is a lipid. Lipids give you long-term energy. So if we look at the structure, it's got these really long chains. You're going to be able to recognize them by these long chains because it's going to take such a long time to break these down. That's why it's going to store energy in our bodies rather than give us that quick available energy like the carbohydrates did. Now let's open this on up. Because these lipids are going to be mostly to store the long-term energy for us, it's going to be things that are going to store inside of our body. So things like fats or oil or even wax. All these things kind of store the energy in our body. If we look at just a cell, the outside part of the cell, remember, is called the cell membrane. That's what we call the phospholipid bilayer, which has the word lipid in it, right? So another example of a lipid we see is in the cell membrane. And that's because oils and waters don't mix, and you don't want your cells to dissolve inside of your solutions. Now, the monomers are going to kind of be a little different. They're actually subunits when we look at a lipid because there's 2 different ends to these. So the monomers are subunits. We're going to call them the glycerol end and the fatty acid. The glycerol end is the short end that's got the C's, the O's, and the H's. So we're going to circle this glycerol end right down here and label it GLY for glycerol. The long chain here, that is the chain that's going to be your fatty acid. That is actually going to be the thing that requires a long time to break down. That's going to give you that long-term energy storage. And that basically wraps up all of our biomolecules.